But let me begin by introducing you to the company and our agenda today. And then as everybody gets joining us, we will go into the meat of the presentation. Okay, so today we will be talking about autoimmune and rheumatic diseases. It's a huge area, it's a huge therapeutic area, so we picked just a few to give you a better feel for imaging in autoimmune diseases, Scleroderma, Myositis, and of course all of them are driven by inflammation. To demonstrate DYNAMIKA today, we also decided to do something slightly different. It's going to be a demonstration of a methodology called DYNAMIKA. DYNAMIKA was invented in the University of Leeds by Professor Francesco Del Galdo, who will join us in 30 minutes time. What's specific about it is that it's an academic invention that was validated and then put as a part of clinical trial on DYNAMIKA. So while by itself it's incredibly interesting methodology, we will show you how to make it real. It's a hybrid read between AI and an expert. And again, that makes DYNAMIKA demonstration and use of this methodology particularly unique. And of course, then we will open for a live Q&A. Again, thank you very much for joining us. So I will begin with introduction of image analysis group. So we are Imaging CRO. We've been founded in 2007, and since then, over 700 trials, which include a lot of the musculoskeletal orthopedic trials, a lot of autoimmune, rheumatic, rare, bone health, muscle studies. All of that is quite relevant to imaging, as you can imagine, because in these studies, you do get to use a lot of MRIs, a lot of ultrasound, X-ray, DEXA, and PET. So as a company, we ensure that sponsors have standardized set of FAQs, processes, study documents. We ensure that the sites are able to acquire high quality images through specific training and as well as additional quality controls. We make sure that the data flows smoothly through our platform DYNAMIKA, which we develop in-house, and you will hear more about it. And once the data is available on the cloud, we deploy quantitative or qualitative read, or sometimes hybrid read, which you will experience today. Lastly, once the data is ready, we export it to a sponsor or a CRO. We operate on 3 pillars. So advanced imaging infrastructure is probably the diamond in our crown. DYNAMIKA, it's a cloud-native SOC 2 iSensor Part 21, Part 11 compliant solution, which is operating system for an imaging CRO, which we deploy in every trial. Shreya with me today will be demonstrating DYNAMIKA. You could see here that DYNAMIKA offers quite amazing functionality in addition to managing imaging data in clinical trial. We provide API to third parties to power up any kind of algorithm, even if it wasn't invented in-house. We allow very fast configuration of the platform for any study, speedy QC of images, and of course, real-time access to information through the dashboards, or sometimes even to the raw images themselves. So from a sponsor point of view, you have control over every stakeholder on the trial and visibility of what's happening. IAG provides end-to-end services as a project management. We have US, Europe, UK, and India locations. We have added operations in China and Australia via our partners. We help to design imaging strategy and select imaging endpoints for the trial. We definitely help you to assess the sites and understand how the imaging protocol and site capabilities work together. And of course, then we provide everything from soup to nuts, from quality control to central review to data management, everything that trial may require. Within IAG, we maintain a network of about 650 scientific experts that includes radiology readers. We work with established reader groups as well as individual readers who are experts in what they do. We handle any modality from X-ray, CT, MRI, PET, DEXA, you name it. DYNAMIKA maintains about 200 read criteria, including some of the very sophisticated ones. We provide additional services in cardiology, safety, and cardiac. I see some comments. So If anything, please do leave a comment on the chat. We will definitely come to them. If anything, I can do, we have an assistant here for the webinar who can also help, hopefully. And just to finish off my slides, I would invite you to join our next section, which will be presented by Mikael Boesen, and to say that today we're showing real examples of something we have done before in our trial. And you can see here highlighted collaborations. So DYNAMIKA is something we deployed in Bergen Equine trial. We will be showing you inflammatory activity published with Eli Lilly. We have great collaborators at Amgen. Again, we will be showing you some of the methodologies we deployed in the phase 4 trial for Penelas, and some of the work on myositis, which was done in collaboration with Merck. We will cover it high level, but please come back and ask more questions, because this is what we're here for. Answer your questions and hopefully help you to run successful imaging studies. Michael, I'll pass to you.
Thank you, Olga. I hope you can all hear me well. As mentioned, my name is Mikael Boesen, Professor of Radiology at Copenhagen University, and I've been collaborating with IAG ever since the foundation. And I would, in the next 20 minutes or so, take you through the, let's say, a short overview of what have been done, and from an imaging point of view, what you need to consider when you're doing your trials. First of all, there's a lot of inflammatory diseases out there where inflammation plays a crucial role, especially in the autoimmune disease realm. And just highlighted here some of the, let's say, the more prevalent ones that are on the market: rheumatoid arthritis, psoriatic arthritis, osteoarthritis, axial spondyloarthritis, as well as some of the autoimmune tissue disease, lupus, scleroderma, dermatomyositis, and also idiopathic myopathies, as well as polymyalgia or rheumatic. And all of these areas, there's no such things as one size fits all. They all come with different challenges and benefits when it comes to imaging, and that's why you also need to design your trials with imaging according to that specific disease and also the specific mode of action of your drugs. And this is why it's so important that imaging is included early on in the design phase, so you can ensure that you use imaging in the correct way, whether it's like enhancing your inclusion criteria, whether it's excluding specific findings, or if it's for efficacy or safety. So all of that is important. Obviously, the images always comes in conjunction with the clinical symptoms. But the clinical symptoms tend to be, let's say, more subjective, even though they're great clinical scores and out there. That is the, let's say, the first rule in, and usually also the primary endpoint in a lot of trials. That imaging can assist, but they tend to lag behind. So when you have response, you can still have subclinical signs of inflammation that continues to drive the disease, which is important to detect, but also it's, let's say, the manifestations can be, in the early phases, can be enhanced by using imaging. The biomarkers, for the wet biomarker side, they are systemic, as well as some of the urine, let's say, outputs. So imaging is, let's say, you should perceive this as the local objective measure of what is happening in the specific target, whether it's joint or muscle or, let's say, area of interest. Whereas many of the other biomarkers, they're kind of a sum of the systemic behavior of the disease. Next slide, please. And then, let's say, depending on what you want to know. So here's the case, kind of an overview of what X-ray, the plain X-ray is the backbone of all the musculoskeletal imaging, except for the myositis and the polymyalgia part. But they are important in all the joint diseases, because if you take it, you see the changes of the structural changes later. In rheumatoid arthritis, it's up to 2 years after that you have had the disease where you can begin to see these findings. Some studies say up to 6 months, but again, it's later than the disease onset because it takes time to make that destruction. But you can also use them to say that what's the status here, and that status has to be maintained by the drug of action. And you also use it for let's say clinical information, also subgroup information like whether you have erosions or you don't have erosions. That can matter in your inclusion, but they also serve as long-term structural progress, especially they're still stuck in many of the regulatory recommendations. So you can't come around the X-ray. It's needed, but it's also valuable because it gives a lot of input also for the, let's say, differential diagnosis. It's low cost. It's highly available. All sites in the world have this. But again, as insensitive and cannot look at the soft tissue changes that you kind of want to, let's say, address when it comes to inflammation, because that's soft tissue changes, whether it's inside the bone as bone marrow edema, or if it's in the joint tissue of synovitis, or in the muscles as a myositis and so forth. The ultrasound is also a very useful tool, but you have to remember it's very operator as well as a vendor dependent, because all of the different vendors, there are many hundreds out there, they have a specific technology behind it, so they cannot necessarily be compared, and they have strengths and weaknesses, all of them, but at least in the hand of experience, they are also variable. So that can pose a problem, but it's good for screening. For instance, if you want to see if a joint has effusion in it, you can use that because it's more versatile. It's easier to apply when you have the patient in the clinical examination room, and that can be an inclusion criteria, for instance. But also remember that ultrasound cannot see inside the bone, so all the ultrasound, let's say, information is stopped by the bony surface. But it's good to see synovitis and tenosynovitis. And if you put vascularity imaging on top of it, it's called Doppler imaging, then you can also see whether something is highly perfused, thus have inflammation in it. Where MRI kind of, let's say, is the reference standard to a lot of extent, because it's less user dependent. It's more reproducible than ultrasound, and then also 3D. So, and it's the only modality that can see inside the bone. So it's kind of the, let's say, gold reference or gold standard in many trials, because it can kind of include all of what you want at the same time, even though it's, let's say, potentially not that, let's say, accessible, and also comes with a cost, obviously. But the fact is that bone marrow edema, let's say, can be, as the potentially only modality, to be visualized and also be monitored. Then you can see the erosions even better than the X-ray. So it's more sensitive for bone erosions, also for detection of erosions that you cannot see on the X-ray, and also it does also the sensitivity of change for that. Synovitis, obviously, that is a let's say, one of the strengths here, because you have inflammation in the synovium, and here it's called also enthesitis. So there's also, which you see on the seronegative arthritis like psoriatic arthritis and ankylosing spondylitis, Wright's disease, and so on. So it is really both a good inclusion criterion, but it can also separate the diseases, see the differentials, rule out some of the high risk groups that potentially have osteonecrosis, or other things that you would not like to have in your trial that can actually deteriorate the outcome of a trial. And then you can use, not only look at the bone, but you can also look at the cartilage. And the cartilage seems to be, let's say, also an important outcome measure in all of the joint diseases, whether it's inflammatory or degenerative. MRI also allows you to do whole body MRI. It's becoming more and more prominent in some areas because you can screen large body parts, for instance, for bone marrow edema and for enthesitis and psoriatic arthritis, as we did in a trial with Amgen. And it also in the realm of sarcopenia it seems to and obesity and weight loss drugs seems to play a crucial role because you can see the effect on both the fat and the liver and the muscle at the same time. It has a high sensitivity and specificity. When you do MRI, and here it's important to understand that if you want to quantify synovitis proper, then you need to give intravenous contrast gadolinium. And as long as you give, let's say, a recommended cyclic gadolinium, you have a high safety profile and should not worry if you have a normal kidney function that there will be side effects of the drugs. When you give the contrast, you then also have the opportunity within the first 5 minutes while you give the contrast to do what's called dynamic contrast enhanced MRI. And it's kind of in a popular term, it's like putting ultrasound Doppler to the MRI image. And it's more reproducible than the Doppler. And it kind of reflects the same behavior. So it maps the vascularity. I will come into that later in the next slide. But and then on top of that, imaging is kind of this is now anatomy and pathanatomy and structure, as well as inflammatory changes. But if you then go to the PET area, then it's metabolic, and here it's complementary to and not necessarily competing with what you can do with MRI, because MRI cannot see the activity of a specific molecule like 18F fluoride NAFPET. It's very good and sensitive for bone remodeling, and then that can be used for visualizing changes in bone, in the subcortical bone or in the bone area. You can do FDG PET where goes where there's a high sugar consumption, and there's a high sugar consumption where you have a high inflammation. So that's also a very good parameter that you can use for that. Use it also in the clinical setting as well as in research in oncology. PET also is a whole body disease because you can, once you're giving the tracer, then you can image the whole body and then you get information from the whole body. But it comes with radiation and you have to put that into your equation. And that also comes with a higher cost as well as some limitations because not all sites in the world have this. So all of this can be handled within the DYNAMIKA platform because it's all of these images comes with a specific, let's say, imaging format. It's called the DICOM, and that so whatever comes in DICOM, DYNAMIKA can handle, and have been applied in various trials, either in conjunction or separately, depending on the outcome and the focus and the primary status of what you are examining, as well as your phase of the trial. So here's an example of an early rheumatoid arthritis. You see on your left-hand side, that's an X-ray of the hand, and that's the right hand. And then it's the same image, the right hand on the side. You see that the thumb is flipped, but that's because the hand is imaged in the posterior anterior, whereas the let's say the MRI is imaged according to the normal and anatomical landmarks. So that's why they're 100 degrees flips. But anyway, what is shown here in the upper row is the baseline image. And this is a usual type of sequences that you use in a rheumatoid arthritis trial. You see the T1 weighted to the left. Here, this is also what's called the fat image. And it gives you anatomical depiction. So it's good for erosions. You can see there are some erosions. You can also see in the wrist bones, there's some dark colors because on T1, fluid in the bone or fluid in itself become darker hypo intense, as it's called. So darker bones compared to the long bones of the fingers that are more bright because they contain fat, because it's the yellow bone marrow. In the middle, it's the stir image, and it's usually popular terms called the water sensitive image. It can be named a stir, proton density FATSAT, T2 FATSAT. All of them kind of reflect the same, different technologies. But in the periphery, you use the stir because it's more robust for suppressing the fat. Whereas when it comes to the, let's say, larger body parts, then you tend to use another sequence. But anyway, on that, you can see that the bones in the wrist, they are bright. Almost all the bones in the wrist, they are bright, meaning that they have bone marrow edema. And we know from histology trials that studies that this represents true inflammatory inflammation inside the bone. So there's some lymphocytes and macrophages, and let's say inside that bone. So it's inflammation in the bone. And then on the right hand side, you see this is a T1 after contrast where you have suppressed the fat, and whatever becomes bright or hyperdense, it's because it takes up contrast. You see that the contrast is taken up in the bones as well as in the surrounding synovium. And that helps you to really see the synovium. And if there were an effusion there, the effusion would be dark and not bright. So you can separate the effusion from the synovium. And on the right-hand side, it's an actual view of the wrist, let's say proximal wrist area. You see the ulna and the radius, and you can see that there's an enhancing lump of synovial tissue there, as long with again dark bones. The lower row, you see that is 3 months after beginning of a biologics, and here you have a imaging response. So the bones in the wrist on the T1 to the left has become more bright, meaning that the edema has gone. And you can see it's the same what you see on the stir in the middle. So many of the bones have not become dark. There's still some small islands and a few bones that still have activity. So you're not, let's say, completely regressed the inflammation. And even though the patient doesn't feel symptom, this is a sign of subclinical inflammation. And you can also see on the T1 on the right-hand side there that you have regressed the volume of the synovium around the bones, and the bones have become more dark again with small areas of spotted enhancement. And on the actual view, you see that here that you have regression of the same area of the proximal wrist joint, where you have reduced the size of the volume of the synovium to more than 50%. And this is how it happened in the early phases of arthritis. When it comes to the later stages, you will become more and more fibrotic. So these fluctuations will not be so apparent. The only thing that will change is the perfusion inside the synovium. Next slide. And this is why, let's say, Olga and I, during our PhD, we co-developed a methodology that could, once you have given the contrast and while you are giving the contrast, could actually make fast images, T1 images around of the same area, and then make a time series over the first 5 minutes of scan time that encapsulated where the contrast went, how fast it went there, and how much contrast every image voxel or image field in the anatomy that you were looking at would take up over time. And that makes some time intensity curve, which is called, and these curves represent in each image voxel the behavior of the contrast over time. And that gives you a lot of possibilities, meaning this is where you, let's say, comes into what means of the ultrasound Doppler analogy, because it highlights the perfusion inside the tissue, and it also sees the perfusion inside the bone, as well as the whole anatomy, whereas ultrasound is more focused and superficial. So this is a true, let's say, great technique to within a certain volume to actually see what is happening. And you can quantify it because it's not static. You don't score it as you will do with the conventional static images as a synovitis score of 0 to 3, for instance, or a bone marrow edema score from 0 to 3. You actually take the volume of what is enhancing, and then you weight it against the slope of the curve, the enhancement curve, or the height of the enhancement curve, or the area under the curve. That gives you a lot more variability, and thus it's also more sensitive to detect inflammation and also more sensitive to show response to treatment. And that has a reproducibility of 3%. And the sensitivity to change less than 5%, and between days in the same patients, less than 5% variation, as well as a static, let's say, perfusion over time in normal. So it really represents that. And when we come to that, then the clinical remission does not, as I said, does not necessarily tell the full story of what is happening. So the subclinical inflammation is important to detect, but it's also important to detect what is happening at the early phases of your trial. So you can, by having a higher sensitivity, you can also reduce your sample size tremendously. But again, it really depends on what is the behavior of the drug. So this has to be applied and also built around what is expected from the drug. And here the earlier phases comes in handy to design that in the later phases. But the clinical finding, you do synovitis mapping, so we can do it in all inflammatory joint diseases. So we also use it in oncology. So it doesn't really matter which disease it is. It doesn't really matter which anatomy it is. It just highlights. As the example here, you can see that this is a patient with psoriatic arthritis and what's called dactylitis. That is inflammation in a finger. It's also called a sausage finger because it swells up and looks like a sausage clinically. And it's inflammation in all tissue. You can see on the stir that the whole third finger is bright. And on the dynamic DCMRI, you can see there's a very high yellowish curve here, meaning that you have high, let's say, activity in the same area, and the corresponding ultrasound Doppler, where you see that you not only have a tenosynovitis, we also synovitis, and the ultrasound show you as well that you have some new bone formation in and around the joints. But so this is the partial, let's say, the clinical reduction in the partial imaging response that you can detect, but it also can show you within 4 to 12 weeks, very fast response, and then it's earlier than the static ones, as well as it can show you inflammation in areas where you don't have a scoring system like dactylitis. So it also gives you possibilities to actually develop some new composite scores around diseases that have been difficult to encapsulate because they have very many different tissues that goes into it. So if you take the next one. And jumping from this, so inflammation is also important in myopathies and in sarcopenia. Sarcopenia comes usually as part of the, let's say, the metabolic syndrome. So it comes with, let's say, sedative aging, but also obesity also drives this. And here imaging can really also be used as a biomarker. And it can be used as a biomarker not only for detecting the disease, but also for monitoring the disease. And it's also show prognostic value, similar to what we've shown with the other methodology that's been used for quantifying inflammation. So this really depending on your question, imaging can serve different purposes. And here you see an example in a sarcopenia obese patient where we kind of can segment the muscle. We can calculate the fat inside the muscle, the subcutaneous fat, as well as the intramuscular fat, and also in the different muscle group, whether it's anterior, posterior, medial, or even separate muscles with automatic and semi-automatic approaches. And on the image on the right is what we also published with Merck. So you can really it's Duchenne's muscular dystrophy, where you can see that imaging not only plays a role of detecting the disease, but also showing the activity. This is a T1 where we segment the muscle and the fat inside the muscle and use that as a biomarker for change. But you can also apply a stir, meaning that you can or give contrast and then map the muscle perfusion, or do T2 maps again that shows you the activity in the muscle that should be reduced. And this is also a regulatory FDA requirement that you use at least the stir and the T1 to visualize activity as well as fatty infiltration. And that can be quantified, and it's inside DYNAMIKA, and it comes with these outcome measures automatically once the segmentation has been done. So the trial design, as I hope you can see that depending on what you want to know, what diseases you work in, imaging has to be shaped around that and designed specifically to that, because every trial has its own more or less proprietary imaging design, but also it's important to take into account the mechanism of action of the drug. So whether it's a seropositive, seronegative, rheumatoid factor, anchor positive, no rheumatoid factor, HBL, HbA7 in ankylosing spondylitis, where they use DAS28, use the Casper criteria, or the CRP, or the SR for monitoring activity in the blood. Then imaging can, let's say, show you something what is happening at the local site. The screening, you use X-ray primarily always when it comes to joint diseases. It has to be there, and then you can add the ultrasound, the MRI, whether you want to screen out or include specific subgroups, endotypes or phenotypes. Then you obviously, on the static imaging, you can do synovitis scoring, tenosynovitis scoring, bone marrow edema scoring, erosion burden, and then put on Doppler or DCMRI for vascularity quantification. And then the structural outcomes, they come either as categorical, or you can also, in some diseases, there is also some where you can map the increase of the bony size, or in bone diseases, map the content of the calcium inside the bone. So the tubercular size as well as the calcium. So either using DEXA or other methodologies. So it really, as you can see here, it's a long list of checking off what you want to have and what you can have in order to answer the specific question that you might have around your mechanism of action, and also depending on the phase that the drug is in. And then coming to an old trial design that's image analysis group been part of lately. So here the phase 4 trial with Amgen, where we applied, let's say, all available types of imaging, whole body MRI, DCMRI, contrast MRI of the hand, as well as muscle quantification and scoring of in diseases. And so we developed here not only a new score for dactylitis, as you can see in the left-hand figure in the lower row. That's an example of dactylitis, how it resolves after 24 and 48 weeks, whereas 93% at week 48 had complete resolution almost of dactylitis. And you can see that how this would look at with DC, but also we have nail beds, as you see on the upper row. Not only the synovitis goes down, but the nail enhancement goes down as well, and that will come as a poster or an abstract and a paper later this year. So again, these are something where we don't have scores for imaging. So you can do the usual suspects, or you can co-develop new modalities that actually encapsulate your inflammation better. And then finally, this is a MoonLake immunotherapeutics phase 2 trial where they use the NAF PET to look at bone activity. And as you can see here, you can see that the activity around the sacroiliac joint, as well as in the bone in general, because it's not only a sacroiliac bone joint disease, also a bone disease. It is completely changed with the treatment. So you have much less activity in the bone. So you're kind of having a larger effect, and you will not be able to detect this if you only was scoring this specifically to a specific entity tissue in a specific anatomic site.
I will thank Mikael for informative presentation, and I could see that Professor Francesco Del Galdo also joined us. So we will allow Francesco to speak about a different methodology called DAVIX, just before we jump into DYNAMIKA live demo of DAVIX. So Francesco, I'll click through a few slides. So hopefully...
Thank you. So good afternoon.
Here we are.
There we go. So yeah, very interested so far. I'm in a clinic environment, as you can see, and actually this is one of the things that I appreciate the most of DYNAMIKA is plug and play from anywhere. And DAVIX, it's a very different type of MRI that we invented here in Leeds. DAVIX stands for Digital Artery Volume Index. Hi, Thierry, good to see you. And so DAVIC stands for Digital Artery Volume Index, and it is an MRI that it was invented to detect digital arteries in the fingers, quantify the volume of flow, and normalize it by the finger volume. Why that is important? So there is a series of diseases where, besides inflammation, there is a narrowing of the arteries that eventually contributes to the clinical symptoms. Scleroderma is one of the most famous and most common conditions that has this as a pathogenetic culprit of vascular manifestation. But this is true also in sclerotic graft versus host disease, in chronic allograft rejection, in pulmonary arterial hypertension, and so in all the other connective tissue diseases that are associated with pulmonary arterial hypertension. So we found that the vascularization of the fingers was reflecting the overall disease activity, and this was published many years ago, showing that patients who end up developing digital ulcers are the same patients that eventually have pulmonary arterial hypertension. So to do that, we have, so to quantify the vessels in the fingers, we went on and using time of flight, which is a contrast-free MRI technology that allowed the visualization of the vessels in the brain, we modified time of flight, and we co-invented with IAG and with Olga this quantitative measure. So here we are able now to measure the arteries in the fingers, and you see here in the third square, you see the arteries in the fingers, simply by detecting the amount of water that moves through the slides, and to normalize this volume for the finger volume, because of course people have different volume hands and fingers, and that would affect the volume of the arteries underneath. The ratio of these two is a DAVIX. Now, this was a very elaborate protocol that required medical physicists and MATLAB until DYNAMIKA came into place and made it possible to have it as almost hand-free, and you will see how hand-free it is. They simply require a validation from a clinician. And so we demonstrated that this volume is actually predictive of new digital ulcers in systemic sclerosis, is predictive of future pulmonary arterial hypertension, in a paper that gained the cover of Lancet now 3 years ago. And since then, this method has been increasingly used in clinical trials that assess, they want to assess efficacy also on vascular activity. And this is, in a nutshell, DAVIX. And we can go to next slide.
So, Francesca, we will jump on live demonstration, I think.
Very good.
So, thank you very much for this explanation. So, on the call with us, we have Shreya. Shreya is a part of our product team at IAG. She will do a live demo of DYNAMIKA, showing you how to set up a study, but at the same time, how to read something as sophisticated as DAVIX. Obviously, not every one of our trial has something so sophisticated, but we thought it would be a great example to show you what actually could happen. Shreya, to you.
Yeah, perfect. Thank you, Laura. Yes, I'm sure you've heard about DYNAMIKA in this whole session right now. And what really is DYNAMIKA? It's a cloud-based platform, which really helps all these different users to collaborate on one platform. So you don't need any kind of installation over here. And it's really the architecture is built in such a way that it's secure, it's compliant, and it's flexible enough to handle any kind of complex workflows that is required for any kind of a study protocol. And so the architecture is built in such a way that it can integrate with any kind of third-party tools as well. So really, DYNAMIKA is something that is built for an enterprise scale level to handle large studies with complex workflows. If you go on to the next slide, I think that would give a little good idea how the workflow really looks like in DYNAMIKA. So it starts with a basic study configuration as step one. And we go ahead and start getting in uploads into the platform. We allow a lot of data sources, mainly DICOM, but we also allow other sources like DEXA, video, photo images, PDFs. And all of these images and documents that come into DYNAMIKA would get quality checked by imaging specialists in our team. They would then be labeled and passed on forward for the reading analysis, where the readers come in and perform the final analysis required for the trial. And finally, we would allow an export as well as required by the client. At the end of all this, as sponsors, we also provide them with the ability to see certain dashboards to understand how the study is progressing, give them some good metrics on it, as well as we have audit trails to show the user actions that are being performed in the system, as well as reports of the readings that were completed. So this basically shows the whole loop that the workflow that we follow in DYNAMIKA. This particular workflow is a more granular workflow for the use case that Professor Mikael Boesen was speaking a few slides back about quantifying myositis. And this actually shows how these different users interact in this workflow, where it starts from the left, where the site user uploads images. Our system, DYNAMIKA, would parse and anonymize them. It goes on to the imaging specialist who performs the quality check. You see the boxes 1 and 2 where they can do some pre-processing that is required. So any kind of segmentations that they can do here, it can go ahead and we pass on these pre-processed images to the readers to finally amend, and they can then sign it off and submit the reading on DYNAMIKA. So it really shows like these different users who interact, but DYNAMIKA allows a single platform for all of them to communicate and for the data to flow through. Yes, I think now is a good opportunity to share my screen, and we can maybe walk through this particular reading criteria. I think I'll start with showing the DAWICS reading criteria that Professor Del Galdo was just speaking about. So I'm sharing my screen. I hope that's visible to everyone. Perfect. All right. So this is a login page for DYNAMIKA, and I'm logging in as a reader role. So I just need my credentials. So as long as you have your credentials, you can log into the platform. I am given an option to select a particular clinical trial I'm part of. So I'll select my DAWICS clinical trial here. And this is a list of readings that is available to me. So as a reader, you would see these lists of tasks that you need to open up. And let me just click on a particular reading task. All right. So what you're seeing on screen is actually the image viewer. On the left-hand side, you see the quality checked labeled images. You see the main image viewer in the center, and you see the ECRF or the reading form on your right-hand side. Now, the DAWICS form had some initial image quality assessments. So you had to select how was the quality of the image, if it's optimal, suboptimal, poor. As you can see, we had to select laterality for the images. So if you had a left hand, a right hand, you select the laterality. Sometimes they were both the hands, so you selected both and performed the analysis in one form. I currently have a left hand case to show here. And as you can see, there are these different boxes here where we are showing the individual digits. So we have a digit 2, digit 3 until digit 5, right? And each of these have the arterial volume and the finger volume to be captured here. So as Professor Del Galdo said, like as a reader, he would select a particular volume to begin confirming the segmentations. And once we select the volume, as you can see, it's kind of pulling the segmentations to show on to the main image viewer. And here we see there is a volume that is being captured here, and this is coming in from this segmentation here. As you can see, we're talking about arterial volume, so it's very small volumes. I'll pause here and maybe let Professor Del Galdo speak a bit more.
Yeah, can I intercept here? Yeah.
Yes.
Yeah, because thank you very much, first of all. And of course, when for us was an incredible big jump to go from a MATLAB complicated protocol to this platform. And what I really valued is that you can verify the area that is automatically tracked missed here on this slide. First of all, you see this is a severely affected hand. We expect to have 2 arteries in each finger, and you see that the arteries here are partially visible on the index finger, partially visible on the 3rd finger, and you can see actually how they narrow in area. And this is very classic of scleroderma, is this patchy involvement of the arteries, and they're poorly visible on the 4th and 5th finger. And this we demonstrated in our old paper. These are the fingers affected by digital ulcer when you don't see the arteries. So here you see the automatic segmentation does a great job, but then you can go on and I don't want to preempt your presentation, but what I was doing as a clinician is say, okay, there is a small area that is missed here, and I have the opportunity either to erase the wrongly assigned area or to, and you can make it as small as 0.5. So the arteries have a maximum diameter of 1.5. So 0.5 is a brush, it's all you need. And here you can track the arteries. And by doing that slide by slide, then I get the volume. What I noticed, and was a nice little attention, is that the color of these dots is the same colors of the finger. You see, the square here is green and this area are green. When you go to another finger, you'll find it pink, and the same dots here are pink. So that allows the clinician not to erase the wrong areas. Sorry for the interruption, but this for somebody on my side of the desk, it's a very useful tool.
Yeah, thank you, Professor Del Galdo. Yeah, so as you were speaking, I was kind of amending the segmentations here. So if you see, I was kind of painting in new regions. And as I paint, what's happening is it is automatically calculating the volume and capturing that in the form here. So as a reader, you can amend these segmentations by drawing into new regions, erasing some existing regions. So for example, I just erase a big bunch here, and we can see it's calculating the volume, and it's reflected a changed volume in the form. So that's how Professor Del Galdo would actually interact with this Davix form and capture these different arterial and blood volumes for every digit. After all the volumes have been captured, you would go ahead and lock the segmentations. So this is just so that all of these values go back into the database. And once you lock it, we can then submit this form. So as part of the submission, we need to provide our credentials again. So this is complying with the 21 CFR Part 11 electronic signatures. So the reader who's performing the reading will have to re-enter their credentials, and they can sign it. Yeah. So I think I'm missing certain segmentations here, but the idea is after it has been submitted, we will then get a report back for the project manager to look at. So yeah, that's basically the reading part for Davix. I think I can move on and maybe show in also, if we have some time remaining, we can also show in how the study configuration and uploads also look like. Yeah.
And as Shreya is setting this up, feel free to drop your questions in the chat. So we'll be happy to answer those. Obviously, you feel free to follow up afterwards, because we see a lot of friends on this call. So, we know that you would have questions because it's specific to your study. So whichever way you would like to follow up. So as you could see, Shreya was just for a second Professor Del Galdo, right? So analyzing the image and looking at it. Now she's going to wear a slightly different hat, a hat of a project manager, and then she will try to do, some of the segmentation, some of the upload, some of the image you see, just as we would do on a normal study. So feel free to drop your questions. She will continue with the demo maybe for another 3 minutes, and then we'll see if we have 5 more minutes for questions.
All right. So I'm logging in as a project manager right now. That's a different role in DYNAMIKA. And this role essentially helps with configuring the study as well as defining the reading paradigm that is going to be followed. So as you can see, I have a list of studies that I'm assigned to. And if I go into any study, we need to give in certain descriptions for the study. For example, some information about the trial, what kind of a therapeutic area, what are some assumptions for the subjects coming in this first and last date as well. We also need to configure some imaging labels, which are these labels that go ahead for the quality check. So the imaging specialist who would come in the workflow would use these imaging labels to label the images that come in, and they would get moved on into the reading workflow. So these labels define what kind of modalities we're going to expect in the trial. We can define the schedule as well. So if you have multiple cohorts, different visit configurations in every cohort, we can configure that as well. And we can configure the forms, that is the EDTF forms that the site user would fill in while uploading images. So that can be configured on a visit-to-visit basis as well. And this is where they would invite the users in the study. So you need to be a part of a clinical study to be able to see anything in DYNAMIKA. So that really ensures that there are boundaries and rules and permissions set for anyone logging into DYNAMIKA. Yeah, this is also a little bit about the dashboards as well. That would be interesting. This is a sponsored dashboard that they would look at to just have an idea about the key metrics, about the key progress of the study. And we could also have a look at these reports as well in the platform. So I'm just opening a report, and this is how they would also view reports here. So yeah, this is basically the study configuration part. Maybe I would also take a minute and show you how easy it is as a site user to upload images on the platform. So let me log in as a site user. This is another role. And I need to select a study as usual. I have a list of subjects here, and to initiate an upload, I need to click on a particular subject. I need to select the type of file. I need to select the visit for which I'm uploading for. I go into the next step. I fill in the EDTF form. So I need to fill in the month and year for the subject. When was this image acquired? Who is uploading this image? Go in next. And I just need to drag and drop my DICOM files. So I have a zipped DICOM file ready with me. So I'm just going to drag and drop it here. And I'm going to upload it. That's it. That's basically how fast it is for any site user to go through these steps and upload an image in DYNAMIKA. And what's happening right now is it is identifying if it's a correct format of the file. It is performing some pseudo-anonymization to take out some patient-sensitive information. And finally, I would get a successful or an error message depending on what I uploaded here. Yeah, I think that's given some good idea on how we use DYNAMIKA, and it's just a little glimpse of more complex workflows that we have already. Yeah.
Wonderful, Shreya. Thank you so much for that. So I see a couple of questions. So maybe Francesca, that's to you. So obviously, differentiation between arteries and veins. As we, acquire the images, we had a great experience in a clinical trial with that. So please share. You unmute, just a minute. You unmute, Francesca.
That is a crucial point. So the time of flight is designed to pick up flow in one direction, and to do that, there is something the radiographer will describe better than me, but basically they can put a stop to the signal. And so basically what I learned is that the slice is depolarized, and then the flow goes in both directions, but you can pick up only one according to a stop signal that you put on one side. So for the hand, the stop signal is put at the tip of the fingertip, and so you see only flow going from the MCP to the fingertip. You don't see the flow coming back, and this way you measure the arterial flow. So that is a critical part of the acquisition protocol, and that's why in this clinical trial we did phantom scans. So to qualify a center, the center, this required a 3 Tesla MRI, yeah? But to qualify a center that has a 3 Tesla MRI, the protocol is shared. Protocols like the sequence is slightly different according to the type of magnet, from Siemens to Philips to GE. and that they acquire an image. The image is uploaded like it was a mock one that is validated. And you can clearly see, because the anatomy of the arteries is different from the anatomy of the fingers. The arteries run only one on each side, very close to the bone. The veins run closer to the surface, and they often have a redundant flow, so they cross each other. They cross each other in the MCP. If you see one, then you can recognize one, and I see the radiologist nodding. So yeah, but that is the so there is something in the sequence that makes it specific for arterial flow.
And I just want to say, you know, it's a real power to have 3 team members on such trial. We had radiologist, obviously, Francesco, who is actually MD, you know, nearly radiologist, but he is a medical doctor, and radiographers who training the sites. I think that's what makes it a huge success. I think we take last question. What is the minimal size of arteries? Francesco, Michael, please speak to that. We can detect on MRI scan. That would be probably very interesting. Then we have to thank you all for attending.
Yeah, we've done, for the part from the Lancet paper, we've done 420 scans, and we've seen it all sorts. The diameter is not much of an issue. What is issue is the velocity. So if the flow is very slow, and this usually happens with the bigger arteries rather than the small arteries. But if the flow is very slow, this may not be picked up, and the sequence needs adjustment. So for blood pressure that is very low, sometimes there may be an issue in detection. But as far as minimum diameter, we have measured up to 0.3 millimeters. Yeah, so a healthy arteries is 1.25 millimeters in diameter in the index and third finger, slightly smaller in the ring and the fifth finger, but we can see up to 0.3. So when we track it with a 0.5, we slightly overestimated. And a healthy ratio of arteries and finger volume is around 1%. So 1% of the finger volume is the artery volume. And we had demonstrated that when it's less than 0.4%, that is an ischemic finger. So yeah, but the sensitivity is more the velocity, and the velocity can be, the sensitivity can be changed by adjusting the repetition time, which is something that is built in the sequence.
Thank you so much. I really hope you feel inspired, because obviously these techniques and these sequences, everything that Professor Boesen and Francesco Del Galdo spoke about, they could be taken forward in other indications and other trials. So if you think of something, please don't hesitate to reach out. We're always very happy to talk about experimental sequences and your ideas. But let me thank all of the speakers and all of the audience for joining us. Please come in April. We're going to have another webinar focused on bone health and DEXA and osteoarthritis, something very different but very dear to my heart. So I thank you very much, and we finish here. Thank you.
Thank you.